Okay, thank you, David. This is Judy Campbell, whom David introduced. And what I'm going to tell you about today is our new startup DNATWO, which is a fledgling company and still so small that we're closely partnering with Caltech, where most of the experiments or many of the experiments are done by people in the labs here. So precisely, I'm going to be telling you about precision targeting of DNA repair to treat cancers. And I'll just remind you that despite the progress that you've seen in treating cancer, there are statistics that show us that without new treatments, if we project until 2040, the number of deaths per year for cancer in the United States is going to rise by 60%. So we think we can help with those treatments by applying our basic science. And we're going to do that through studying or using DNA repair. And so if we go to the next slide, I'll tell you what DNA repair is. So we're going to have an introduction here. So your DNA is the most stable molecule in your cells, but this is pretty remarkable because DNA is actually undergoing damage every minute, every second in your cells. And this damage causes problems with the function of DNA and makes it unstable. So what the cells have done is develop repair pathways that remove the damage almost as soon as it goes in. And so it maintains, these repair pathways maintain the stability of the DNA. An important point to remember as we go through the talk is that there are multiple repair pathways for each type of DNA damage. That is, there is redundancy in repair pathways. That's an important point. Could we go to the next slide, Ken? Okay, so now I want to tell you what kind of DNA damage is important in cancer. But you already know that cancer cells, a hallmark of these cells, is that they are rapidly proliferating. That means they are rapidly copying or replicating their DNA. And what we know, having studied replication and repair for 40 years, is that rapid replication leads to rampant and increases in DNA damage. And that damage has to be repaired before the cells can go on and divide and form tumors. And so therefore we proposed, shown on the cartoon on the left, that DNA replication is actually the Achilles heel of the cancer cells. And we were very fortunate because in our work in the lab, we had discovered a protein called DNATWO, which you'll recognize as the namesake for our company, DNATWO. And what we showed in characterizing DNATWO over the years was it is a really special DNA repair protein designed specifically to repair damage at the replication sites. So we proposed then that we should be able, by inhibiting this repair protein with DNATWO inhibitors, to kill cancer cells. So what I want to leave you with is the idea that there are many DNA repair proteins in the cells. So the zip code for DNATWO is definitely DNA repair, but importantly and distinctively, the street address of DNATWO is DNA replication. So DNATWO becomes the Achilles heel of DNA replication. Okay, so the way we plan to use this is shown in the next slide, and it is a concept out of genetics called synthetic lethality, and it's a strategy for killing tumor cells, but being relatively safe for normal cells. And it works like this. So normal cells, because of those extra DNA repair pathways that I told you about, are insensitive to DNA repair inhibitors. So if you treat with a DNATWO inhibitor, you'll see over on the left that a normal cell is perfectly viable. By contrast, if you look at a cancer cell, you see that when treated by an inhibitor, that is lethal. And that's because not only do they have this excessive DNA damage, they're actually defective in certain DNA repair genes. So what we are doing is creating lethality or making synthetic lethality in the cancer cells that have a mutation in this DNA repair pathway by inhibiting the second inhibitor, the second repair pathway with DNATWO inhibitors. And this is the success of this strategy is supported by the use, by the success of another DNA repair inhibitor called PARP inhibitor that many of you may be familiar with. Okay, so having gotten to this point, we decided to form DNATWO, and we as the team shown on the next slide. And so I'll start off introducing myself a little more precisely. So when we realized all of these facts about DNA repair and cancer, I made the decision that I would like to turn over a new leaf in my studies, which involved up to that time basic science, and now try to translate those studies into an actual cancer therapy that could be used in patients. And so having studied all these things for 40 years, this was a turning point in my career, but I was really motivated to do it because I was, first of all, very excited by the challenge. And secondly, I was pretty confident that if the science was with us, we could actually translate it with the knowledge that we had. But most importantly, I'd like to tell you that what I really wanted to do at this point in my career is to pay back agencies like NIH and places like Caltech for supporting me in this basic research over all these years without asking for any repayment. And so I've been doing this as a professor of biology and chemistry, and now I'd like to turn the slides over to Brian to introduce himself, and then Ken. Brian.
Thanks, Judy. I think that what Judy's saying about the details of cancer biology and repair are incredibly exciting. And I think for many of us, this area of oncology and cancer is an incredibly personal issue and area. And for me, 10 years ago, that became incredibly personal when my mother passed away. And it was personal from the standpoint that it was very challenging because I was standing on the sideline and could not really do much. For the first 12 years of my career, I was involved in complex molecule synthesis and the development of new chemical reactions. I'm a professor of chemistry and I specialize in synthetic organic chemistry. And I had spent quite a bit of time in that first 12 years consulting with the pharmaceutical industry, big and small companies, working on many different therapeutic areas, but truly doing that from the standpoint of the engine that drives those areas, which is medicinal chemistry, aided by synthetic chemistry. But when it became personal, I decided to make a real career shift, which was to try to think about ways that my science could translate and potentially be applicable in these areas that I felt so helpless in, namely cancer biology and oncology. And so about that time, I really made a conscious decision to move more toward the areas of translation. And as a result of that, one of the companies that I've been involved in and a founder of is something called 1200 Pharma. For those of you who have ever been to Caltech, you know the street address of Caltech is 1200 East California Boulevard. So this is an absolute Caltech startup that was founded out of Caltech technology. And when Judy approached me to tell me about DNATWO some years back, I got incredibly excited because this would be another opportunity to be able to be involved in moving an area that I felt so helpless in. In this case, it's a different kind of enthusiasm and excitement from my standpoint, because this is really the excitement of working on a brand new target in an area that is so important. And so when Judy got me involved, I was happy to jump in and really enthusiastic to do it. And it's been a fun journey, and I think there are many more years of advances to come. And so I'll turn it over to Ken, our CEO, to give his side of it.
No, thanks, Brian. And I think you're right on. There's kind of a common tapestry, if you will, between the three of us that makes us go after new, exciting things. Very similar. My career started on a traditional path, biology, chemistry degree, go to medical school, then practiced emergency medicine. But about 6 years after that, into practice, I hung up my stethoscope, started looking for a challenge, and joined McKinsey & Company, working in their healthcare practice to help the world's leading pharma companies, solve their hardest and most complex business challenges, from research and development strategies, all the way to product launch and marketing. And I think those 5 years, plus getting an MBA under my belt, kind of created a business platform that was, for me, it balanced what up to that point had been purely science. Since McKinsey, I've been the Chief Strategic Officer at Medtronic Diabetes, for which the artificial pancreas system won Time Magazine's Invention of the Year award when I was there. And then also, I led business development and innovation at Halozyme, taking a small cap biotech all the way over $4 billion through a series of deal making transactions for a drug delivery platform. So, with about 7 of the blue chip pharma companies. And so, that's been my journey up until this time. And in summer of 2019, we formed a roundtable here in Los Angeles that asked the question, how do we make Los Angeles no longer the biotech flyover city between San Francisco and San Diego? You may have seen it published in a trade journal, and Amgen and Kite and Vita Ventures, UCLA, City of Hope, and everyone was there, except actually Caltech. And I have to admit, when the entrepreneur in residence of Caltech, Helen McBride, introduced me to Judy and Brian, what I've discovered is you have just a fantastic ecosystem as a now serial entrepreneur who takes drugs that are first in class, sometimes even first in pathway, into the clinic. Just what you have in an ecosystem is incredibly exciting, whether it's Freeflow or other Caltech affiliated venture funds, or just kind of the system and the benefits that are known as being a private university. So yeah, when we met right in the middle of COVID, virtually, we formed our company, started our mission, and actually raised our funds as well virtually, getting our first 2 million working on a Zoom platform like this. So it's been a fantastic journey, being at the tip of the spear of innovation.
Okay, thanks. And thanks for your enthusiasm, guys. Okay, but now we have to return to basics, because I need to tell you a little bit more about the mechanism of action of DNATWO, so you can understand the milestones that Brian and Ken are going to present to you later. Okay, so DNATWO not only works in DNA repair, it also works in DNA replication itself. And so if you go over on the right, you'll see a molecule that's replicating, DNA replicating all the way over on the right, Ken, if it at the top. Yeah, that one. Okay, so there are intermediates in replication that need DNATWO to get past, because they are intermediates in difficult to replicate regions. So DNATWO plays a real role in DNA replication. In addition, as I've told you, however, DNATWO is a DNA repair protein that fixes really serious damage that occurs during replication. So you can have damage shown in the middle figure over on the right, Ken, where you've got these toxic intermediates that don't even look like DNA, or you can get to the most oncogenic type of DNA damage when replication forks collapse into what's called double-strand breaks. And DNATWO does this all directly, just as an enzyme, but it's a really multifaceted enzyme. So it's an ATP motor-driven nuclease, as well as an ATP motor-driven helicase. And it's these activities that are critical for the functions that I described over in the models on the right. In order to go a little bit, I have to go a little bit more deeply into DNATWO, and that's shown in the next slide. Now, don't panic. This is just a 3D structure of DNATWO. And what I wanted to show you is all of those activities are encoded in different parts of this protein. So you see the helicase that I mentioned, the nuclease that I mentioned, DNA binding sites, and there are protein binding sites all over the molecule as well. So what this does for us in trying to get inhibitors is provide us with many druggable sites, even more than the 3 that I mentioned in the bullets. And what that offers us is the opportunity for many shots on goal here, to actually identify inhibitors. And furthermore, it offers us a diverse pipeline of inhibitors that, because of the different activities, may function in multiple different cancer indications. At the same time, the diagram is supposed to show you that DNATWO is a huge protein, and it's made up of moving parts that have to precisely move with respect to each other during catalysis. And what that creates for a person trying to develop a drug is the problem of expressing the protein in high quantities, which you need, and in expressing it in an active form. And at DNATWO, we have solved those problems with great effort. We also have done a lot of genetic analysis, which we can do in the future based on this structure. Okay, so with that knowledge, then we can go on to the next slide. And we have also profited from the explosion of genomic information from studies of cancer that have been published over the last 5 years. And what I'm showing you here is that DNATWO is clinically relevant. So if we look over on the right, we see what is called a Kaplan-Meier curve that plots the survival of many cancers, 1,000 in 10,000 in this case. And if you look at the red curve, you'll see that cancers that have high DNATWO levels have poor prognosis. So their survival is much lower than the blue line, which are a collection of 5,000 cancers that have low DNATWO expression or lower DNATWO expression. We can dissect these cancers further and look at specific types of cancer, and that's shown over on the right. So here we're looking at a greater difference in survival with breast cancer cells. And you see the high levels of DNATWO are really significantly deleterious compared to the low levels of DNATWO in the upper curve. Okay, so we have additional information from these public databases from which we draw this information, and that's shown in the next slide. Now, these are data, and this is called a waterfall plot, and it was an experiment carried out at Novartis. And what they did is to genetically remove DNATWO from 369 cancer cell lines. And those cancer cell lines are represented by the bars that are shown in color here. And so what they looked at after getting rid of DNATWO is which cancer cell lines could grow without DNATWO, or how did removal of DNATWO affect their growth. Over in the right, you see things that grow better when you have DNATWO mutated or removed. In the middle, you see ones that grow about the same. And over on the right, you see the cancers that we're interested in. Those are the ones that are dependent on DNATWO. They don't grow well when you remove DNATWO. Now, these are 3 solid tumors. And if you don't have time to look at the legend here, I'll just summarize that these are osteosarcomas, colon cancers, and a special type of breast cancer called triple negative breast cancer, or TNBC. And what we're going to tell you about later is our studies on TNBC. And we've actually repeated a study like this with these cell lines, showing they are sensitive to our inhibitors. So now I can get to the nuts and bolts of what we have actually done. So when you are isolating inhibitors, the first thing you have to do is a screen of large libraries of compounds. And we have done that. And shown on the left is what we call our testing funnel, which is a rigorous test of the chemical, biochemical, and biological properties of these compounds that go down in the funnel till we get to a compound at the bottom that convinces us that we have compounds that might work in reducing tumor growth in vivo. And so we got down to 3 compounds, and Ken's going to tell you what we did with those compounds. Okay, so I'm going to turn it over to Ken and then Brian.
Great, thanks, Judy. Yes, so we've built a real comprehensive funnel, as Judy said, and after taking thousands of compounds through, in the end, we had possible chemical matter candidates to take into in vivo experiments. So the question is, when you go into in vivo, where do you want to go? And this was from a scientific decision making. And this was informed not just by the funnel, but also the data that Judy had previously shared from the waterfall chart, from seeing DNATWO as expression, making it a poor prognostic indicator in breast cancer. So we ended up with 2 of these triple negative breast cancer xenografts. I like to call them twins with differences. One was HCC 70, and the other is DU 4475. And both of them are triple negative breast cancer. One grows quite fast, so 1,000 cubic millimeters in a week versus 3 weeks. They also varied somewhat when it came to D2 expression. And then you'll notice HCC 70 is PARP resistant. And that's what we have in this graph here, is you can see HCC 70, this is from the lab that performed the experiments, but the PARP inhibitors, which Judy had mentioned earlier, that work in DNA repair, here in this gray line had absolutely no real tumor growth inhibition, for this cell line. So we think of it as being resistant, to PARP inhibitors. The data cards turned over, and a little bit of a drum roll, look like this. So looking at these 2 tumor models, just briefly in terms of our methods, the triple negative breast cancer cells were implanted subcutaneously in the shoulder region of nude mice. And when the tumors reached approximately 200 cubic millimeters, and 10 per group, were randomized into the vehicle or into the treatment groups. On the left, what you see there with HCC 70, remember this is the so-called PARP resistant xenograph. You see fairly impressive tumor knockdown. Gemcitabine was used as the positive control. The vehicle is in black, but you notice what we were after is proof of principle, finding anti-tumor activity. And all three of the chemical matter scaffolds, showed that, as you can see in the red and then the two gray lines. The other part, though, that's worth hammering home is the tolerability. Remember that Judy had mentioned in her normal cells versus cancer cell slide, normal cells tend to be spared some of the toxicity of DNATWO inhibition. And if you look at this bottom line, the body weight changes that were seen in the mice, remained at least for 372 and 001 less than 1%. Which tells us this was a well-tolerated compound, with the mice. The other one that we looked at was DU 4475. So this is basically a freight train of triple negative breast cancer. It grows extremely fast. Notice that the scales are double and the time is half, right? So this is a fast-growing, hard-to-treat cancer. There actually was not a positive control that had been validated by the lab for this, but the good news is we found with our scaffold 001, it created 100% tumor growth inhibition. In fact, all 3 scaffolds, showed anti-tumor activity, which is promising when you're in a drug hunting discovery posture like we are. So just to sum up, and we have it here at the side of the slide, all scaffolds, showed significant anti-tumor activity. They were able to give tumor size regression, even where PARPs were unable to do so. And then, of course, we showed lots of tolerability when you look at the body weight changes in the mice. So pretty exciting, seeing all the science kind of line up for the in vivo experiments. Now you may ask, well, what about patients? You know, that was in Brian's bio. You know, it's all about the patients. And overall, kind of where we're heading is a promising market and with a high unmet need. I mean, breast cancer is the most common cancer among women and has an addressable market of over $20 billion. Now, there are therapies for several breast cancer types, or I should say subtypes, which in the past, a diagnosis would have been paramount to a death sentence. And these have been transformed into chronic diseases at this point. That said, the in vivo models that I just presented earlier for triple negative breast cancer, that's a very aggressive form of breast cancer. Conventional breast cancer therapies like Herceptin have limited effect. And in the case of triple negative breast cancer, even though it represents just about 12% of all breast cancer cases, it contributes to about a third of all deaths. Now, to make matters worse, triple negative breast cancer tends to occur in younger women, meaning before age 40, and also in disadvantaged populations, such as Black women for whom access to healthcare can be a challenge. So our real headline here and why we're pursuing innovative, first in class, first in pathway is these drugs are needed for new targets are really going to be required to meet that challenge. So that's where the excitement comes from and gets us up in the morning. So with that, I think I'm going to turn it over to Brian for where we've gone to since then with the violin chart.
Yeah, thanks, Ken. So what Ken showed was an incredibly exciting proof of principle experiment of an in vivo efficacy study. That said, the molecules that we used for that study, we investigated and found them actually quite challenging to prosecute further. These molecules, in some senses, suffered from some hallmarks of medicinal chemistry, things like solubility, and also the ability to differentiate from a structure-activity relationship standpoint. It was hard to move those molecules further. And so what we decided to do as an alternative, and something that we've been building since we started, was the development of a truly high throughput screen that would allow us a fluorescence-based assay to be able to perform many thousands and even potentially hundreds of thousands of screens simultaneously, so that we could amass a large data set and begin from a much better standpoint, from the standpoint of chemical matter and structure. So the development of this high throughput assay is another major milestone for the company. And it's shown schematically on the left. Essentially, this is a readout of nuclease activity, so one of the functions of DNATWO. And it allows us to screen through, again, many thousands of molecules very rapidly, in this case, in a 380-well format, 84-well format. We did this recently on a screen of 30,000 molecules over the course of a couple of weeks, and we obtained all of that data. What was great about this is that we selected molecules from a highly curated samples set. So there are many CROs that will provide you with hundreds of thousands of molecules, but they're not all created equal. So we tried to curate these from the standpoint of a deep experience that we have at DNATWO in medicinal chemistry space from molecules that we thought would be reasonable starting points for medicinal chemistry. We ran that screen, and the results are shown on the right-hand side of this slide in what we call a violin plot. Essentially, what this shows is the integrity of the screen is terrific. It shows that actually the majority of molecules have no effect on DNATWO, which is actually a good thing, because we want molecules that are highly selective and highly specific and efficacious eventually for the inhibition of DNATWO. We don't want to affect anything else in a cell or in our bodies. So what's good is that out of this 30,000 screens, we had really what we think are about close to 500 actual hits, which is a pretty tight window. There were a few thousand additional ones that we could potentially think about following up on later. But it's really those initial 474 hits that we think are the most important. And importantly, that comes in a variety of scaffolds of chemical matter. So shown on the next slide is really a map of the chemical space of this 30,000 compound library. So if you imagine defining the differences between molecules in a variety of ways, you can describe them in lots of different ways. We use computation. We have a computational expert on our team to separate those molecules into what we call chemical space. So this is the entirety of the universe of potential of how molecules can fit with relationships to one another. What you'll notice is that many of these stars are very separated from one another, and others are clustered together. So that shows that in our 30,000 compound library, some molecules were very similar to one another and others were very different. What's also shown here on this slide are the differences in activity. So strong inhibition are marked in green, weak inhibition or no inhibition is marked in red. And so you'll see that some of the outliers are individual green dots and other outliers are individual red dots. That's fine. We can prosecute those or investigate those further. But what's truly interesting here and where we can really use computation immediately are in these clusters. And so, this blow up at the bottom of the slide is really a cluster of a number of molecules that were within this 30,000 set for which we have real inhibition data. And what you'll see is that although the molecules are close together in this cluster, they actually have differential activity. That's a great sign because that shows that small changes in the chemical structure can actually have real significant changes in the inhibition in the assay. And we think that's a great place to start because that means there will be relationships to the activity based on the structure. And that's where medicinal chemists love to live. And so that's really the next stage of what we're going to do is to start to prosecute these hits, the ones that we think have the best promise first, and we can broaden accordingly from there. But we'll move through that testing funnel that Judy and Ken were talking about very succinctly as we prosecute these hits further and eventually get back to what we think will be excellent chemical matter for moving forward for a drug.
I want to start out with a question for you, Judy, because I think it's just really interesting to kind of go back and think about when you first, decided to pursue this research direction, and perhaps, when you had maybe your first aha moment along the way, and then how that led to you actually, if you could provide commentary on when you actually said, I want to do a company around this, because that translational piece is something that, we're obviously trying to have more scientists take that step, but you decided to take that step. So if you could maybe contextualize some of that journey for a little bit, I think that'd be interesting to hear.
Well, Caltech has traditionally been dedicated to basic research, especially in the biology division. So when I first realized that I might be able, with my knowledge, to guide the translation effectively and talked about it with my colleagues, they were really, oh, Judy, you're selling out and everything. And that kind of galvanized me into, okay, I don't think I'm selling out. I'm going to show you. And so I decided I'm going to found DNATWO, because if I apply to NIH for more grants to do this translational work to the normal NIH, not to the small business program, they're just going to say the same thing my colleagues here at Caltech said. So anyway, that's the history. The other thing I wanted to say is that I don't want to be immodest, but I had some confidence that I had enough knowledge after all these 40 years of studying DNA replication that I could use that knowledge and really make the translational work, if anybody could. That is, if the science was really going to allow us to do it, that I could help guide that translation really effectively. And I could do, but I needed other people, right? I'm not a chemist. I couldn't isolate the molecules. And so I was thrilled with Brian. And I also didn't know anything about business. So we were extremely fortunate to get someone as prominent in LA as Ken, and not only to keep the lights on, but to make sure DNATWO would be there if we finally got this science done. So that's, is that what you're asking? Yes.
Yeah. And then, along that path, you mentioned Helen, and I think it's really interesting to reflect on the, I don't know, the ecosystem that's building up around Caltech, and just the, you know, the resources that are available at Caltech now to help people out. Maybe you could comment on, and maybe that involvement, and just how that played out. And
Yeah, so ever since David Baltimore came to Caltech, he brought a guy from MIT that transformed our so-called technology transfer operation. That built gradually, but about, I don't know, let's see, 10 years ago, it really became formalized, and they introduced this position of entrepreneur in residence. So they hire some successful entrepreneur to come in and to start interviewing Caltech professors to see if they had anything that might be translatable, that they might not even realize was translatable. So that entrepreneurs in residence are really critical if you want to, and have made the path easy for professors, and which is important because, as I told you, you get resistance. Used to get resistance at Caltech, but now this Office of Technology Transfer, and in our case, it was particularly Helen McBride, who was the entrepreneur who was galvanized by the idea of DNATWO. And so she is extremely well connected in LA. She's a biologist, by the way. She has a PhD. She is my biology consultant in this so far. So she is now a consultant for DNATWO, and remains an important link in the program. But to your point, it's this well-developed Office of Technology Transfer. So they handle our patents, they handle our connecting us with investors, they mediate everything so that we as professors can still run our labs, and we can do this. And that, I don't think it, well, some of the founders have even given up their labs and actually gone to their companies, but most of us don't want to do that. And so there is this mechanism, which is, I think, quite unique to Caltech, that they want us to do both. And historically, you have to know that Caltech was founded way back, or the biology division at least, way back in the late 1920s by the Carnegie Foundation. And they, being from New York, were arrogantly positive about thinking if they brought this very good basic science to Southern California, they could kick off good medical schools here. And so that is just a little Caltech history. So now we're just realizing that history decades later. And of course, the money. The Caltech actually has an innovation fund that you can apply to get started. And it has investors like you at Freeflow, and the people at Wilson Hill who provide the money. And of course, the money is not easy to come by, but it's much easier to come by here at Caltech. So I guess I forgot to mention the obvious.
So David, maybe it'd be helpful to have Brian sort of come in on top of this, just having founded a company previously, Brian, and just sort of comment. You touched on this a little bit earlier, just comment on the evolution and the differences that you've seen, having done this once before, and now, you know, here you are again, 2023. What are your thoughts?
Yeah, I mean, I think everything that Judy mentioned is critical, and I think it has been building over some time. You know, in the more on the chemistry side of the institute, you know, I have had colleagues that have been very entrepreneurial since I arrived, which is now, you know, almost 23 years ago. And so actually one of the first interactions I ever had was with the technology transfer office. And they were, you know, just very upfront about, you know, being enthusiastic about protecting the intellectual property of the Institute, and we came up with a plan for how we would pursue that over the decades now. And we've been doing that, you know, steadily for a long time. And for a long time, they were taking me on faith. that any of the things that were coming out of my group had anything to do with an actual viable path to licensing. And they floated that for a long time. But then, like I said, they also helped finally, I think, build a culture, starting with this entrepreneur in residence program, where it just became much easier and less mysterious, and frankly, less frightening for a faculty member to dive in. And so that, for me, was really what got the inspiration going for 1200 Pharma. And it was that entrepreneur in residence at the time who really launched that company. And now I've seen it happen again with Judy's technology and with her deep knowledge of DNATWO, that all, as she said, was built on just fundamental curiosity-driven basic research. But these things that we do at Caltech, they're important, and there are real applications to them that, frankly, if we don't do it and we're not the ones driving it, either it won't be done the right way or it won't be done at all. And more often than not, it's simply just not done at all. And I think that is a real loss frankly, for humanity. And so I'm incredibly grateful that Caltech has recognized this and has set up systems that do make it just easier and more straightforward to go for it. It's not easy by any stretch of the imagination to actually make anything work in the real world, but it's certainly not feasible at all to be able to just do that running out of an academic lab. I think we need these kinds of mechanisms to be able to translate this work, and it's been really fun. I mean, I think it's one of those things people said you'd get hooked, after you do it a few times, and I guess I'm falling into that category at this point.